instrumentation, this 3' sulfur containing molecule was not cleaved at all. And so this was a disappointing result because it had taken many weeks in the fume hood to synthesize this variant molecule, and we had expected that it would be cleaved more quickly, and instead it was not cleaved at all. One possible reason that something might not be cleaved would be if it didn't even bind to the catalyst. But we had an independent way of analyzing binding, shown in the table here, and you can see that the 3' sulfur containing substrate had a binding constant that, this is expressed as a dissociation constant. So in fact, this is about 2.5 times tighter, more tightly bound than the normal substrate. But the main point is that it's bound to a similar extent as the 3' oxygen containing RNA. So that wasn't the explanation. And yet we had this very large amount. If we strained our eyes and used sophisticated instrumentation, we could detect a little bit of cleavage, but it was 1,000-fold reduced from that of the normal nucleic acid substrate. So at that point, Pitcher really added just a dash of a different metal ion, manganese, to the reaction, and all of a sudden the 3' sulfur containing substrate was cleaved quite well, either in the mixture of magnesium and manganese ion or in manganese ion alone. So the ability to cleave this slightly different substrate was restored when a different metal ion was added. Well, what could that mean? And we were reminded of some early work by Mildred Cohen and also some studies done in Mo Cleland's laboratory, where the preference for metal binding was compared, the ratio of coordinating of a metal ion to oxygen relative to sulfur, which of course was the atomic switch that we had made. Magnesium ion has a huge preference, 31,000-fold, for binding to oxygen relative to sulfur. Manganese is much less fussy and prefers oxygen by only 158-fold. So the explanation then was that there must be a metal ion situated right next to this atom that is part of this bond that is being cleaved in the ribozyme reaction. And that if this atom is oxygen, then magnesium works fine in this site. But when you change it to sulfur, magnesium no longer is effective because it has such a huge preference for binding to oxygen relative to sulfur. But now manganese, swapping manganese into the metal binding site can restore the cleavage. So the idea is that this ribozyme recruits a metal ion from the cellular environment and uses some of the catalytic power of a metal ion to help cleave nucleic acids, a strategy that is very much analogous to that of many protein enzymes. Why does this make sense? Well, here's a picture of the so-called transition state of this reaction. I described this yesterday as the halfway point of the reaction. This dotted line is a bond that's half formed at this instant in time as the guanosine is trying to make an oxygen-phosphorus bond at the cleavage site. Remember, it becomes attached to the intron during this reaction. And the other bond shown here is one that's half broken. And as this bond is half broken, there's a redistribution of charge. Charge builds up on this atom, and only by having a metal ion close by can you stabilize that developing negative charge and thereby speed the reaction along. We think this accounts for about 1,000-fold, or 3 orders of magnitude, of this 11 orders of magnitude of catalysis that we have to explain. Other features such as orienting the attacking guanosine at just the right angle so that it can make a very efficient attack on this chemical linkage is another catalytic strategy that helps speed the reaction along and helps account for that very large rate acceleration. So we now summarize in this more complex diagram, now putting back the normal oxygen atom in the RNA substrate and the more normal cofactor magnesium ion, some of the interactions which help stabilize this halfway point or transition state in the chemical transformation and together contribute to making the reaction occur on the time scale of a small fraction of a second as opposed to 100 years. Now, you can see that the ribozyme surrounding the reactive groups has not been shown in any detail here. It's just shown as a shaded surface, but you can see that that shaded surface is critical in aligning all of the other players at just the right angle to make an efficient chemical reaction. So the next natural question might be, what is that shape of the RNA catalyst? How do you build an active site composed of RNA? RNA is composed of a series of helices, and it's quite easy to understand how you get to that first order of folding. It has to do with A pairing with U, G pairing with C. It has to do with hydrogen-bonded base pairs, very similar to the ones that hold the double helix of DNA together. What is more difficult for us to understand is the question of how do you pack those helices together into a very specific folded entity, which is then able to arrange the substrate molecules in just the right way. So it's this higher order of packing which is the subject of a lot of current investigation. One way that you might think of looking at the structure of an RNA molecule would be electron microscopy. And we've done this in collaboration with Yuhua Wang and Jack Griffith at the University of North Carolina. And the results are at the same time encouraging and frustrating, just like most of experimental science. So the encouraging part is that you can see very discrete globular molecules. And I've been claiming to you that like a protein enzyme, this RNA would have to be globular rather than strung out like the DNA double helix in order to have an active site cleft and act like a catalyst. So it looks globular. The frustrating part is that you can almost imagine seeing some regularity in these globs, but the resolution of electron microscopy just isn't good enough in order to be able to pick out specific helical elements within that globular ribozyme or RNA catalyst. And even if you go to higher magnification, you can see some interesting substructure, but you can't quite pick out individual helices. So you need a marker. You need some way of identifying one helix from another. And staring at these pictures, it struck me that perhaps there was a fairly simple solution to this problem. That if we could just, and let's go to tRNA as an example, transfer RNA also would be much too small to visualize its detailed structure by electron microscopy. But if we would extend this helical portion of the molecule out to the right and then extend this helical portion downwards to make sort of reporter arms that would be large enough to see in the electron microscope, and then counting on the fact that perfect double helices of nucleic acid are quite rigid rods, we should be able to monitor at least the angle between these two. For transfer RNA, using this as a model system, we know that that angle should be about 90 degrees. So we extended the two arms of transfer RNA by 70 base pairs each, looked in the electron microscope, and lo and behold, the transfer RNA itself is this little blob where the two arms meet. You can see there's no detail visible there at all, but you can see the two arms extending, and when you measure the angle between the two arms, you can see that it centers at 90 degrees. And then it has some distribution. They aren't all exactly 90 degrees. Some of them, here's one you can see, is a little bit more of an acute